A very good morning to everyone. Let's crack CSIR UGC NET together. And that is the name of our channel. Let's crack CSIR UGC NET together. We are doing a crash course for GATE 2021 in the evening, and we are doing a crash course for CSIR UGC NET in the morning. I'm your educator Farida Johar Bandukwala. I have a teaching experience of 10 years. I've qualified MP SLET examination in year 2018. You're opting for Unacademy, what all things you are going to get. You're going to get daily live classes so that you can be in that momentum of studies. And in the live class, you can ask your doubts and queries directly with the faculty member. Then the whole syllabus is quite structured. As you know that there are 13 units in the syllabus of life science, so you need to be very precise and specific about which topic to pick up and study. So the whole syllabus which is taught on an academy is very much structured so that you can have that wise decision of which topic to be studied and in what depth, right? Then live tests and quizzes are taken so that you can evaluate yourself that how much you have learned so far, and unlimited access will be provided towards all the videos and study material available in whichever category you are subscribing for. Unacademy tries to give as many free live classes as it can. These free live classes are conducted on two platforms. First is YouTube. So presently you are watching my class on YouTube. It's a completely free class. Another thing is a special class. For attending the special class, you need to download the Unacademy Learning app from the Google Play Store. Then you need to make your account. After making your account, you'll be asked to provide the referral code of an educator. You can give my referral code FB01 and you can avail the facility of attending the free live classes. By using my referral code FB01, you can attend my classes as well as you can attend the classes of other educators as well. Unacademy has got one more platform, and the name of that platform is Plus Platform, and this Plus Platform is a paid platform. And on Plus Platform, all the 13 units are discussed in detail. Each and every topic is covered, and for all the upcoming classes, crash courses and short-term courses are conducted so that you can always prepare for the upcoming exam. And the prices are very low. For 12 months you need to pay 11,500 rupees, and for 24 months you are going to pay 16,800 rupees. And after that much discount also you can use my referral code FB01 and you can get an additional 10% discount. So do not forget to use my referral code FB01 and avail an additional discount of 10%. So let us begin with the topic, that is NMR. Nmr spectroscopy. The full form of NMR is nuclear magnetic resonance. We are going to talk about this topic under following heads. We are going to talk about introduction. We are going to talk about basic principles in NMR. We are going to talk about instrumentation. We are going to interpret the NMR spectrum. Then we are going to talk about the applications. These all points will be covered. So coming to the introduction and history. Introduction and history of NMR. So it was first of all described and measured by Isidore Rabi in 1938. 1938. Isidore Rabi in 1938, first time. After that, in year 1946, another scientist, that is Felix and Edward, in year 1946, they refined the technique for use of liquids and solids, for which they shared a Nobel Prize in year 1952. So it is important to know that they have got a Nobel for this in year 1952. After that, the observation of resonance frequency of a molecule allows a user to discover the structural information about the molecule. So these two things are coming under history. And now what is NMR, why it is used for, so that we are going to see. So the observation of resonance frequencies of a molecule allows a user to discover structural information about the molecule. So the words which you need to remember in this page is resonance frequencies, and it tells you about the structural information of a molecule. Structural information. So resonance frequency tells about the structural information. Now we'll be talking about the basic principle in NMR. So principle. So first of all is magnetic phenomena. So what does magnetic phenomena mean? And because of what these magnetic phenomena is actually brought. So it is because of the atomic nuclei. This magnetic phenomena is because of atomic nuclei, and these atomic nuclei, they work as tiny magnets. They work as tiny magnets. After that, the magnetic phenomena, first of all, atomic field or atomic nuclei itself works as tiny magnet. An external field will be applied to align or to oppose the normal magnetic field of the atomic nuclei. That is the tiny magnets which is present in the molecule itself. An external magnetic field will be applied. So second is external field. And this will actually align or oppose the tiny magnets. Align or oppose. Now next is number of nucleons. Number of nucleons. So what do we mean by this? So the number of nucleons, the number of magnetic fields present in the molecule. It must be odd for residual magnetism. Must be odd for residual magnetism. For example, must be odd for residual magnetism. So let us see one of the example to understand this. Like talking about hydrogen. Hydrogen exhibits the magnetism, but deuterium doesn't exhibit this thing. So hydrogen exhibits, but deuterium does not. Right? So magnetic phenomena, then number of nucleon. Then the third thing which we need to know in the principle is resonance condition. Resonance condition. So what do we mean by this resonance condition? It's a transition from low to high energy state. So transition from low to high energy state occurs. Then either a constant magnetic field is applied, an appropriate region of radio frequency is swept. A magnetic field will be applied and radio frequency will be swept across it. So either a constant magnetic field is applied and appropriate region of radio frequency is swept. After that, either a constant magnetic field is applied and appropriate region of radio frequency is swept, or a constant frequency is employed and external magnetic field is swept, vice versa of the first condition. So constant frequency is employed and the external magnetic field is swept. External magnetic field is swept. So we have seen three things, that is magnetic phenomena, number of nucleons, then resonance condition. Then now we need to see what is a chemical shift. So chemical shift. So chemical shift, what is it? From where does it arise? So it arises from the applied field inducing secondary fields at the proton by interacting with the adjacent bonding electrons. That is the chemical shift, and it is measured relative to internal standards. Okay, so chemical shift, it arises due to external applied field which induces secondary fields at the proton by interacting with adjacent bonding electrons. Adjacent bonding electrons. So that is the chemical shift. The normal bonding is going to be changed. The external field will let the protons interact with the adjacent bonding electrons which are available for bonding, and it is measured relative to an internal standard. After chemical shift, we need to know what is spin-spin splitting. Spin splitting. So spin-spin splitting is actually seen in high resolution spectroscopies. Seen in high resolution spectroscopy. Right? So we have seen so many things regarding the principle, that is, we'll just revise, that is magnetic phenomena. Then we have seen number of nucleons. Then we have seen what is a resonance condition. Then we have seen chemical shift. And then we need to know about spin-spin splitting. So these 5 things come under the principle of NMR. Now talking about the diagram of NMR unit. diagram of NMR unit. How does it look like? So there will be external magnets. Ignore the shapes. These are the external field. This is the sample tube. On this sample tube, there will be application of radio frequency as well. Radio frequency. So this is the radio frequency output unit. There will be radio frequency input unit. So I'll just use another color. So this is the radio frequency input. And this blue one is the radio frequency output. Okay. And this is the field. Okay. And this is our sample tube. And these are the external magnetic fields. External magnetic field. So this is how, and here there will be a spectrum that will be obtained. It will be in the form of certain kind of graph. This type of spectrum will be obtained. Okay, so this is the diagram of NMR unit, though it is looking quite simple over here, but it is actually a quite big apparatus. And now we'll be talking about the interpretation. Basically, interpretation is important when it comes to any kind of technique. So interpretation. Interpretation of NMR spectrum. So first of all, number of main signal is equal to number of equivalent protons. Number of equivalent protons in unknown compound. In unknown compound. So number of mean signals is always be equal to number of equivalent protons. So that will be the guess of how many protons are present in a compound. After that, chemical shift. So chemical shift is going to indicate, chemical shift indicates the type of hydrogen atoms. The type of hydrogen atoms. So if hydrogen atoms, they are present in methylene, or methane or any kind of methyl groups. So that will be easily understood with the type of chemical shift it is bringing about, right? After that, spin-spin splitting. spin splitting. So spin splitting is basically arrangement of groups in the molecule. Arrangement of groups in the molecule. That how they are actually arranged. After that, talking about the area of peak. Whether it's a narrow peak or whether it's a broad peak. So talking about the area of peak. So number of hydrogen nuclei present, number of hydrogen nuclei present in each group. Present in each group. So for example, relative area of methyl peaks in the propane would be 6:2. But in butane, it is going to be 6:4. So example, relative areas of methyl peaks in propane would be 6:2. And in butane, it would be 6:4. That is how the peaks are calculated, and that is how interpretation of how many carbons are there, how many hydrogens are there is done with the help of NMR. So we have talked a lot about the interpretation, instrumentation and principle. Now we need to know that what are the applications. So applications in the field of biology specifically, because there are applications in the field of chemical sciences as well, but we are talking in terms of application in the field of biology. So first is molecular structure determination. Molecular structure determination. So first, most of the work is done in the solution form. Work is done in solution form. Then drug metabolism studies and mechanism of action of drug is also done through NMR. So drug metabolism and mechanism of action. Drug metabolism and mechanism of action can also be studied with the help of NMR. Then low resolution NMR on lac repressor was also done, and head repressor headpiece and BPTI was also done with the help of NMR. Then high resolution spectrum of NMR is also done for inflammatory proteins, then plastocyanin, then epidermal growth factor and interleukins. So high resolution NMR high resolution NMR spectrum on structure of various things such as interleukins, then plastocyanin, etc. After that, solid state NMR is also there, and it is done for Alzheimer's beta amyloid peptides. So solid state NMR done for Alzheimer's. After that, it can also be done for understanding the molecular behavior in solution. The second is to understand the molecular behavior in solutions. Molecular behavior in solutions. So results of studies of protein folding are exemplified by nucleases, then cytochrome C, then barnase, lysozyme, ubiquitin, etc. So that is the molecular behavior can be understood through this. Then enzyme kinetics, very important. Enzyme kinetics, example for chymotrypsin, papain, then pepsin, ATPase, etc. So molecular behavior can be understood with the help of NMR. Then next thing, phosphate metabolism can also be understood. Phosphate metabolism. So for phosphate metabolism, P31 isotope, it exhibits the nuclear resonance. So that will be used. The isotope 31 exhibits nuclear resonance. So changing the concentration of AMP, ADP, ATP in the living cells, the phosphate metabolism can be understood. Intracellular and extracellular inorganic phosphate concentration can also be understood through NMR. So changing concentration of AMP, ADP and ATP in living cells. Then intracellular and extracellular inorganic phosphate concentration can also be determined through NMR. Then for nucleic acids, its application. So next, fourth application is in terms of nucleic acids. So structural studies of DNA and RNA can be done through that. Structural studies of DNA and RNA. Then drug interaction with DNA. Then interaction between the binding protein and DNA. Interaction between binding protein and DNA. Then interaction between protein and lipid bilayers. Interaction between protein and lipid bilayers in membrane. These all things can also be understood with the help of NMR. After that, some of the applications would be related to the medical field. So another application is in the field of medical science, that is MRI. MRI is magnetic resonance imaging. Magnetic resonance imaging. We have done a video on MRI in detail on the same channel. So if you have that curiosity, and it is quite important according to CSIR examination, so you can go and watch the video. So in MRI, resonance of proton is considered. Then a proton is more sensitive. Proton is more sensitive. Then it is present in abundance. because in human body or in any organism it is present in abundance. So what all things are done through MRI? First is human brain imaging. Human brain. So in human brain, imaging of posterior fossa, brain stem, spinal cord can be done with the help of NMR, that is in the field of biological science. When the technique is applied, it is called as MRI. Then detection of demyelinated lesions, hemorrhages, then differences in the water content in white and gray matter in brain and between the normal tissues and tumors is done with the help of MRI. Then second, MRI of abdomen can be useful in understanding the metabolic liver diseases. In adipose tissue, the proton signals from lipid is measurable and hence fat deposit, fatty liver, that those all things can also be understood with the help of MRI. Then what are the advantages of MRI? Advantages of MRI: it limits the risk of X-ray. X-ray, uh, the need of X-ray is eliminated, though it is not much clear through what I have written. So let's write only in a systematic manner. Eliminates risk of X radiation. Then excellent and contrast resolution. Excellent and contrast resolution. Then detecting the diseases at a very earlier stages. Detecting disease at early stages. These are some of the advantages of MRI. Now, when we have seen the advantages of MRI, it is compulsory for us to know the advantages of NMR as well. Though it is the topic, so it is but obvious thing. So NMR advantages. So first is considerable amount of information can be obtained from a single analysis. Considerable amount of information can be obtained from a single analysis. Then this technique is non-destructive at all. Non-destructive technique. Then replicate analysis can be performed. Replicate analysis can be performed. Then precious samples can be recovered even after NMR spectroscopy. Samples can be recovered as well. So these are some of the things of NMR spectroscopy. Let us just see what all things we have done. So we have covered all the points which we were destined to. So history says that first time Isidore Rabi gave the idea of NMR, and in year 1946 Felix and Edward, they refined the whole thing and they got a Nobel Prize for this in year 1952. So if you're saying NMR, what does it tells about? The resonance frequency of any molecule is enough to know about its structural pattern. Then in principle, we have seen the magnetic phenomena. One is the inherent magnetism of a molecule, and one is the external field. The number of nucleons, how many number of nucleons can be made. Then resonance condition, either a constant magnetic field and variable radio frequency or a constant frequency and variable external field can be used. Then chemical shift is whenever there is a external field which is applied, the protons, they are going to interact with the adjacent electrons. So that is the chemical shift. And then we have seen the whole structure, that how it looks like, external magnetic field, then radio frequency input, radio frequency output, and those all things we have seen. Then how to interpret the NMR spectrum. So it is done under following things. So number of main signals are going to be equal to the number of protons present. Chemical shift tells you or indicates you about the type of hydrogen atom. Then spin splitting is going to tell you about the arrangement of the groups in the molecule. And lastly, the area of peak, the number of hydrogen nuclei present in each group, is understood with the help of the area of the peak. Then in application, we have seen that molecular structure determination, then molecular behavior in solution can be understood, then phosphate metabolism can be understood, then nucleic acid structure, interaction, all those things can be understood. Then in medical field, MRI is completely based on NMR. It is done for human brain studies in various kind of tumors. Then in abdomen, the fatty tissue which is deposited on liver, the fatty liver can be easily identified with an MRI. And what are the advantages of MRI is there is no risk of harmful radiations of X-rays. Then excellent contrast is there between the resolution, and it detects the disease at a very early stage. Those were the advantages of MRI. Now considering the advantages of NMR, so considerable of amount of information can be obtained from a single analysis. Just a single analysis is more than enough. And it's a non-destructive technique, and replicate analysis can be performed and samples can be recovered. So like this, we have completed NMR in a detailed manner. And as we say in the starting and end, let's crack CSIR UGC NET. And that is the name of our channel as well. If you haven't subscribed our channel, subscribe our channel. Hit the bell icon for getting all the notifications of the upcoming videos. And if you really like my video or you have gained something out of it, give it a thumbs up. It really motivates the educators like us when we see many likes on the video. And lastly, use my referral code FB01 for getting the plus subscription, and you will be getting a 10% discount. And you can also attend the free special classes by using my referral code FB01, so that you can you can get the benefit of attending my special classes, which is live or which is already recorded, and you can attend the classes of other educators as well by using my referral code. So this is it for now. We'll be meeting again at 5 p.m. and discussing some topic of GATE syllabus. So we'll be meeting in the evening, and tomorrow 10:30 and 5 p.m. So do not forget to be there for the class, and till then keep on studying and be happy. So finally, it's a bye from my side.